Okay, welcome everyone. Welcome to BIOHOUSE. My name is Shai Meltzer. I'm the CEO of BIOHOUSE, and we're here at BIOHOUSE Hadassah on a festive opening event of going back to real events. Continuing our very successful collaboration with Pharmaseed, which has been cut by COVID-19. We're happy to be back with professional events for our professional biomedical community. BIOHOUSE is going on its 4th year now here at Hadassah, building what is essentially Israel's largest biomedical community on ground. We are here today with BIOHOUSE Hadassah space that has been opened for almost 4 years now. And 2 years ago, we opened BIOHOUSE Labs, letting in biopharma companies, early stage biotech companies that were very various, brought a very big variety in the types of project they brought into our labs, which was a good validation point for us, opening a platform that was flexible enough to bring in companies doing microbiology, cell therapy, gene therapy, clean tech companies doing chemistry. So we're very happy with the mettle of companies working at BIOHOUSE Labs, and we thought it was a good opportunity to set our opening an event of this season with Pharmaseed on cell therapy. At BIOHOUSE, we put a very big emphasis on tech community. We connect the entrepreneurs that work at our spaces, both at Hadassah and the lab spaces, not only to the medical center in which we operate, this case Hadassah, but we also connect to medical centers in the rest of Israel and all around the world in order to bring the connection to the people using the technology at the end of the day to the people developing at early stage, so that they can avoid all the haphazard mistakes that are made during development that bring us back to the blueprints, too often, too many. And we take these types of opportunities to bring people within development to share their insights, to share how their processes are going, and you'll hear from companies at different stages today, so that the community can benefit from being together, which is a big thing of what we do here at BIOHOUSE, because sharing the space are companies that are in different stages, doing different types of projects, but the people, and the most important part of these companies are the people, have been doing many projects for many years, and so they have a lot to share. So not only is the space itself shared by the different companies, but also the experience and the know-how. And that for us is essential for the success of a place like BIOHOUSE. And that's why for us, collaborating with a CRO like Pharmaseed, which has many, many years of experience and know-how in cell therapy and in other areas, as you'll hear, for us, it's essential to bring as much know-how into the space that we don't hold internally, which is part of why we also involve in BIOHOUSE outsourced consultants. We bring in experts from all fields relevant to biomedical development to come and consult, speak, confer, make these types of events, one-on-one meetings, and we also connect the companies to mature industries. Many of the companies here at BIOHOUSE have been connected to late-stage pharma, late-stage medical devices, digital health companies. IBM runs the Hadassah Accelerator on the top floor here at BIOHOUSE Hadassah, and the team at IBM is very, very open to meeting all BIOHOUSE members, and this is something that we were very keen on when we set out with this new brand and this new model. So coming back to this event, working at the space with the people that are working here and people outside of BIOHOUSE, working with a CRO, bringing all this knowledge from the outside, and doing all of this just walking distance from the Hebrew University and Hadassah Medical Center is key to how BIOHOUSE does business. And we take this festive opportunity in this event to tell all of you here today and all of you online that you are more than welcome to come and be in touch with all these people that you hear today and with us in order to see how we may help you connect from afar or from here on close and other places that we may be operating, because this is the tip of the iceberg. This is just a small example of the types of activities that we have here, the types of people that we work with, and the opportunities that we help facilitate are endless. We're very happy to finalize This is actually the last week, or next week is the last week, of a program we're doing with the Korean government. We've been working in the past 6 months with Korean startups, helping them enter the European and U.S. markets, helping them connect to the Western world of biomedicine. And it was a very good opportunity for us to benchmark everything we know with the companies we work with an ecosystem that is not accustomed to working in the standards that you all here work by on a day-to-day basis. So you can all be proud of the fact that you're actually at the front of biomedical development, and other people in other parts of the world can learn a lot from you. And that's something that we make sure that happens. So that for me is just an example of how we focus our efforts for our members here at BIOHOUSE, as Pharmaseed does with their clients. So to finalize, I want to welcome all of you here today. It's very refreshing to see all these good old friends and many new faces that came here for the event, and welcome all the people online to hear this refreshing symposium on cell therapy. And I want to welcome Galit Ginsberg from Pharmaseed to say a few words as well. Galit, please.
Thank you. Good afternoon, everyone. I'm going to make it short because we have a long agenda and very interesting one. So I just wanted to start with a story. How many of you know how cell therapy actually started? The very, very first recording. So I'm going to tell you because I made my homework. So it started in the 19th century with a neurologist at the age of 72 who wanted to fight aging. And the way he did it was to take, let's say, testis juice from monkeys and inject it to himself in order to, as I said, rejuvenate his sexual prowess. So that's the very, very beginning. But since then, medicine has, sorry, I'm a bit excited about this event. So medicine has evolved from herbs, utilizing herbs, spells, blessings, and chemicals, to cell-based therapy, gene editing therapy, and let's say organ replacement. And the future is still unclear, let's say. And at Pharmaseed, we are an organization of scientists who want to be at the center of the revolution of medicine. And we are very, very motivated and excited. I don't want to talk a lot about Pharmaseed. You will see that our speakers today had some of their projects, developing projects with Pharmaseed, and you will see some of the results. We have some companies who reached clinical phases with their even cell-based therapies. And before we start, just a quick, very, very quick thank you, first of all, to you for joining us here, for taking the time to join us here in Jerusalem and online from all over the globe. To BIOHOUSE for hosting this seminar here and for a great collaboration, and for the speakers that you will hear today for taking the time to tell us a little bit from their experience and share with us their remarks, their insights, as you will hear far ahead. So I hope you will enjoy this as much as it is exciting and enjoyable for me. I'm coming from master and PhD in cell therapy, so I'm very excited about this field. And let's start. So our first speaker is Mrs. Chaya Mazuz from Orgenesis. Let's welcome her.
Okay, so I want to start by thanking also BIOHOUSE and Pharmaseed for having us here today and for asking me to speak. Okay, so today we're going to speak, at least my presentation is going to speak about personalized medicine, that's autologous products, and it's a journey. Like everything we do in life, it's a journey. The journey starts from the patient, and it goes, as you can see, we have a lot of stops on the way, and it ends back to the patient. So we have two main journeys. The first journey that we have is the supply chain that we just saw. It starts from the patient, and it ends when we reach back to the patient. The second journey that we have is to bring the product from the bench to the patient, just to reach the patient. So we have two journeys. I'm going to focus a little bit on the second journey from bench to patient, and the main presentation will be on the supply chain. Okay, so this is an opening remarks. I think all of us will agree that cell and gene therapy are complicated. It's constant learning. What we knew yesterday is not what we know today, and not what we're going to know tomorrow. The scientific discovery is very fast. There's a lot of great ideas running around, and we want to bring them very quickly, and we want to translate them to the patients. But we need to remember that there are product processes that need to be manufacturing. We need to do them, and not to forget the analytical technologies that we have on the way. And we need to scale the manufacturing. If we have something in the bench, like anything like in a university or in academia, we have a lot of small-scale manufacturing, and we have to have the ability to be able to bring it to a higher or a more reducible scale. Always, it's a balance between speed and the needs of the development of the product and whatever comes with it. And the last remark is long time frame from collection to treatment. It's more a philosophical issue. When we take the starting material from the patient, he's, let's say, 20 years old. When we give it back to him after days, weeks, a month, is that the same patient? Is that his disease progressed, went backward? It's not exactly the same patient, and this is something that we always need to consider when we talk about autologous or personalized cell and gene therapy. The person that we took the starting material from, his cell or tissue, is not always the same person. The patient that we will receive it will get it back. Okay, so now I'm going to go a little bit fast through the CMC issues. So it's very chemical manufacturing and control. It's complicated for me to speak with the mic in my hand, but no, not for you. It's it's okay. So just to say that CMC and quality go together. Chemical manufacturing and control.
Where's that terminology from?
That's the terminology. That's the quality section that you need to provide for the documents. Okay, so we have a variability in the starting material. When we say starting material, it could be any cell, any tissue from patient. We have a high variability of our raw materials. What raw materials do we want to use? Are they human? Are their source human? Are they animal source? They have a very high variability. We have the manufacturing is complicated. It's very manual. It's very small scale because that's how it reaches to us. And last but not least is the issue of qualification and validation. How do we qualify and validate a product that is autologous and it's personalized? How do we do that? I don't have answers for everything, but for most of the things you will have answers today. So when we want to start the manufacturing, when we want to start our development, what do we need to consider? So first we need to consider, what do we do? We want to work on a CAR T, let's say. Where do we get the starting material from? Do we use it from healthy donors or we use it from sick patients? What do we do? The reagents for the cell and for the cell selection and growth, or anything how do we test this? Do we test this on healthy? Do we test it on a sample that we get from a patient? Does that mean if we took one donor, is that enough? Do we need 3? Do we need 5? Do we need 6? What is the magic number here? When we do our development process, how we translate our development to GMP. And GMP is, at the end, mandates our work. And of course, not to mention that we need to do media fills in order to demonstrate aseptic processing. So What does that mean? If we have a manufacturing process for a month, do I need to do a media fill over a month? That means that the company or the facility is going to do media fills all the time. So there's a lot of considerations. And one example is sterility testing for a fresh product. What do we do? How do we validate these things? So let's go back to the patient's journey. So the patient journey, it starts that a cell is tissue or a cell or tissue or cells are collected from the patient. They're transferred then to a centralized manufacturing facility, as of now at least, in most of the treatments that we know. Cells are treated. Now, when they need to go to the manufacturing facility, do they need to be cryopreserved? Do they need to go through any early treatment? Maybe they need to be washed at the site yet, at the clinical site. Maybe they're cryopreserved there. And then they're sent. They could go by truck, by plane, by bike, swimming, whatever. But they need to reach the manufacturing facility. Once they reach the manufacturing facility, they need to be manufactured. And in the meantime, don't forget, we do have a patient in the picture that a lot of times the treatment requires for conditional treatment. Let's say he needs to go through lymphodepletion or anything. So we need to not forget these two angles. So we have two main issues that we want to consider. One is the time that from the patient, when it left the patient, it could be a cell or a tissue, anyway, starting material, till it reaches the manufacturing facility. And the other thing is we call it vein to vein or needle to needle, when we want to see the whole process when it leaves the patient and comes back to the patient. So what would be the perfect order? How would be in a perfect life? And we will take the sample or tissue or cell from the patient. Very quickly, we'll do whatever manipulation we need, and it will reach the manufacturing facility. Doesn't matter what time. People are waiting there, standing, incoming material, SOPs, everything, and we start manufacturing. And the manufacturing is successful. However, what happens if the manufacturing, we have issues? What happens if we have a deviation in the facility? So if we manufacture a regular product, we have time till we finish manufacturing. We could start investigation. We could start talking about it. Here we don't have time. A patient is waiting for us. So we could not accept a batch for many reasons. It could be because of facility. It could be a failure to meet the final spec. So the main issue here is that being able to give the patient a safe and efficacious product is our main goal, and we have to do everything to meet it. I put here two slides about chain of custody and chain of identity, because these are things that are crucial in autologous personalized medication, and we need to know them. So we have chain of custody and we have chain of identity. So what that means, the chain of identity means that there must be a clear identification between the cells or the tissue that were donated by the patient and the batch that the patient will receive. Doesn't matter how it does, if it's by letters, by numbers, but there must be a clear course of identification. The second thing is the course of custody. That means that everything needs to be documented. Who did what? Who did the collection? A lot of times, who's the truck driver? I don't think who's the pilot, but everybody that takes part in this manufacturing from vein to vein needs to be documented, and we need to know. One doesn't go without the other. Okay? If we have chain of custody, it goes only with chain of identity. They need to go together. Okay, so what are the next steps? How do we approach these things? So the main thing is flexibility, and we need to be flexible. And we must learn how to work with risk-based approach. Everything in this world is risk and a benefit. What is best and what is worse. And we always need to use these skills to make these decisions. On a day-to-day basis, all of us do it on a day-to-day basis. But this needs to be documented, and it needs to be done in the right way. So there is, in the GMP guidance, there is already an attitude towards decentralized manufacturing. You're allowed to manufacture your product near the patient. It could be in a point of care. It could be in different ways, but you are allowed to do decentralized manufacturing. So we have the flexibility. We have the idea what is needed. We do not know how to do it. And here we need to, the regulatory challenges are extremely high. We need to reach the approach how to do it. Now, I'll say something in Hebrew, but there's a phrase in Hebrew that they say, Yashan mipnei chadash totziu. That means take out the old when the good comes. Now, this is a term of prosperity. That means when something new comes, because it's a very good times, then we could take out the old for the goods. In our specific, we cannot. That's the fundamental. The old world is the fundamental for this new world that we need to build now. So decentralized manufacturing means that we are able to manufacture in a facility, in a point of care close to the patient. That means that the whole chain of supply, of the transportation, everything that we saw needs to be limited. We have the ability to now to do the manufacturing at one site. And this site could serve one hospital, 2 hospitals, 10 hospitals. Since we live in Israel, the distance is not so big. But this is the main thing of decentralized and point-of-care treatments. That means that we have a new framework, that we don't have to send the product. We don't need to treat it. We don't need to treat the starting material. The starting material could reach us as a fresh product, and we could immediately put it into manufacturing, finish the manufacturing, and based on our stability data, based on 24 to 48 hours, we could give it back to the patient. We will not save the time of manufacturing because that needs to be done, but we will be able to have a more viable product, reliable product, and we could save a lot of issues that may happen on the way. Okay, but what are the challenges of a decentralized manufacturing? So if I manufacture in 20, 50, 100 different sites, is it the same product? Because I want to manufacture the same CAR-T. I want to manufacture the same TIL product. I want to give the patients the same product even if they're in India or they're in Manhattan or in Jerusalem. How do we do, and this is a regulatory challenge, what are the qualifications, what are validations that will be required to show the authorities that we really are manufacturing the same process? Thank God we're able to do that. So this is Orgenesis' approach. What Orgenesis wants to, is presenting now is the point of care, is the ability to manufacture a cell product, a cell or gene-based product, all over the world in the same manner and give it to the patient in a timely manner. What does that mean? What our aim is first to make the manufacturing process automated, as less open manipulation as possible, and then to do in a centralized way to do qualification and validation, and then only to transfer a product to these point of cares that could be all over the world. However, we do have challenges, and what are the challenges that we could see? How do we control this? So having a control site, a lot of us did already clinical trials. Think about a CRO. Think about a CRO that runs clinical trials. They run clinical trials all over the world, but they have one headquarters, or like maybe 2, maybe 3. And then when the agency wants to come and do a pre-approval inspection, they do not go to 60 clinical sites. They will come and select what sites they want to see. So what is a central site? A central site or a controlled site, it doesn't matter, it's a different terminology. Just you should know, it's not something that I made up. The MHRA already has a white paper, the FDA started discussing it, the EMA is already moving forward to this, so we understand that this is the right approach. So what will this central site, we will have sites manufacturing these point of cares, decentralized manufacturing all over the world, and we will have a central or control site that will have an oversight. That means change management, deviations, CAPA, trend analysis. Everything needs to be escalated to this control center, and they will do all the coordination. And if they find that something that happened maybe in a site in Kathmandu, maybe has an impact on the site that is working in north of Israel. So the main issue is that the The control will be only in one, it could be maybe regional, but the concept is to have a control site. And they will do the oversight, as I said, and in countries that we need qualified person from a phase 1, this will be they could delegate it, but the oversight release will be done by a centralized qualified person. So this control site needs to do the training, needs to be responsible for whatever we just discussed. I need to move forward because I'm going out of time. So I didn't know that Shai is going to say what he said, but this goes very well together. We need to work together. This is the new world. We are working together for years in the cell and gene therapy, and we don't need to change it now. And we need to work together. That means that the challenges that are facing Orgenesis as a company faces every other company. The bottleneck of autologous manufacturing is not something that is local in Israel. It's a worldwide issue, and we need to address this together. We need to put everybody together. And in the past, before COVID, we did this a lot. It stopped a little bit, but we need to go back. And only if we work together, we will make this happen. Okay, I'm finishing because I'm getting my cue. So I didn't put all the entities that need to be together, but these are ropes. And each person and each entity is a rope. But if we do not tie the ropes together, this is not going to happen. So we need to put forces together. That means industries, CMOs, that do have a lot of experience in centralized manufacturing. We need to work, all of us together. Only working together, we will make this world much better, and we will make these therapies accessible for all. So usually I want to say thank you, and this is my name. And usually I just want to, this is open. When I joined this industry 20-something years ago, there were a lot of good people on the way that always I had a question. Even if it was stupid, I could have picked the phone and asked them a question. And I'm happy to say that I'm happy to be this person for any other company. So if you ever need me, you could always just lift the phone, ask a question, send an email. I would be happy to help like I was helped in the beginning of my way. Thank you very much. Questions? Everybody's good, I gather?
So we have a question from the Zoom participants. What are the current regulatory demands of preclinical toxicity tests for CAR T cell therapy targeting solid tumors?
Okay, that's not a quick answer. But we do need to do tox study for, it's nothing to do with my presentation with the supply chain, but yes, we need to do, we have to do preclinical toxicity studies for CAR T. Now, based on the vector and the hinge, you need to decide what will be. Again, this is a risk-based approach. You need to decide what data was already published, what is in the literature, what is the similarity to other products, and then decide exactly what needs to be the extent of your tox studies. Ooh, wow, okay.
At the end of the day, risk-based approach would basically dictate that it would depend on the tumor, depends on the disease, depends on what else is available, depends on the vector in manufacturing. And in the spirit of collaboration, I've done that exact work with Pharmaseed. My company allows me to collaborate and consult for those issues. And the answer would be that if you're going after something like glioblastoma with a brand new product that has very low risk, you can get away with, maybe just a 30-day NSG study. Whereas if you're going for Hodgkin's lymphoma with something that a brand new virus, the demands would be much higher.
Thank you, Uri. Yes, please.
How optimistic are you that it will be possible to do How optimistic are you that it will be possible to do point-of-care manufacturing for personalized cell therapies and to be sure that the quality control and quality assurance will meet the specifications for the product across the globe?
Thank you. So first of all, I'm a very optimistic person.
I'm happy to hear it.
Okay? So first of all, we're there. We're already there, and we're already discussing these things with the regulators. So this is already, it's not something in the future. We're there. We're starting negotiation. We're starting to present the idea. There's a lot of industry groups that want to support this approach. So we're working together to reach the regulators, to discuss. A new manual will need to be written. New guidelines will need to be written. It starts from everything, as I showed before about the qualification and validation, but it also, what are the requirements for the point of cares for these manufacturing facilities? We are not manufacturing in this humongous CMO facility. So what is really required? I had here a discussion before with Uri. We need to do things, not because we can, but because we need to do them. So it's not the easiest way to say, okay, let's sterilize something. No, is this really needed? So we're there. I think we still have a way. I hope till I reach my pension, it will be already a story to tell, but we're there.
A lot of companies from the stem cell therapy are now also working with exosomes and extracellular vesicles. And my question is, do you believe that the regulatory aspects or the pipeline working with exosomes and extracellular vesicles would be somewhat easier compared to what was with the cells and the stem cells, or would it be exactly the same and this field will need to be matched what was before?
I think one thing that we're learning as we do not match any more therapies. Each therapy is a standalone, and we need to find the right way, but we do have the fundamentals. And I do think, I don't believe in something easier or less, because I think everything that we want to address, we know how to address it. I think it's not one of the hardest things that we have on the table now, and I think it's doable. And I think we should learn from what was done already. There are products that are approved, and you know what? It's a good thing to learn from products that failed, from products that did not pass the regulatory approval. We need to understand why. Why are these issues so critical for them, for the regulators or for the patients? That's the main thing. So I think we can learn, but everything is, I feel that every day is a new day. Every day we learn something new. Okay. So thank you very much.
So, thank you, Chaya. And we'll move on to our next speaker, Uri Herzberg from, now he works in Cabaletta Bio. Before that, he works in Cellularity and Celgene. So he has a lot of experience in cell therapy.
So, thank you so much. And actually, without asking permission, I expanded the scope of this discussion from cell therapy to novel biological agents. Reason being, if you look into what we're doing right now in cell therapy, the vast majority of products are autologous. Out of those autologous products, the vast majority of those products are T cells, which we really understand very well, specifically because of some groundbreaking work that was done at the Weizmann Institute by Zelig Eshar and his collaborator at Penn, Carl June. And specifically what we're doing is we're transducing those T cells mostly with viral vectors. We're putting them back into the patient. It creates a nightmare, as Chaya would attest to. It creates quite a few challenges. But then, take that just as a case in point, there are investigators already in the running that are saying, we don't need to do this transduction. We don't need to do this shipment back and forth. We can do the transduction in the patient
we can inject an agent to the patient that would transduce those T cells in the patient, would do what those T cells do without having to go through the supply chain. Now, is this still a cell-based product? I would argue that it is, because the mechanism of action of this product is still cellular. So this is now called a novel cell therapy because this is still inherently very different from aspirin or from Tylenol, because the action is through the cell. Take another case in point. Another way to activate those T cells, the way that those T cells work is we engineer them to put some sort of moiety. The moiety sees the tumor, and then the T cell kills the tumor. That moiety can be a single chain, antibody. That moiety can be some sort of, TCR that would recognize the tumor. Now, other investigators are saying, okay, if this thing works, why don't we create an antibody, bispecific antibody, that on one arm would bind to the patient's own T cell, and the other arm would bind to the tumor that we know. So now, instead of going through this back and forth shipment engineering, we would create bispecific antibodies that would do the work, that are easier to produce. But again, is this cell therapy? And again, I would still argue that this is still cell therapy because the mechanism of action, the way that you kill the tumor, is through the activation of a cell. So being that this is the case, we come to, I cut my teeth in drug development at Johnson & Johnson and partly at Celgene. We did mostly small molecules and biologics. And the first thing that the clinical director would ask you when you come up with the idea or when you come up with the program was, what is your PK/PD? What happens to the drug when it sees the human? What happens to the human when it sees the drug? Without PK/PD, you do not know how to dose your drug. And I would argue that in cell therapy, we still don't have PK/PD. We don't know where the cells are going to. We don't know how they die. We know that they expand. Because once they see the tumor, the T cells do what T cells know how to do when they see a target. They expand like crazy, which is good. Which basically, some of them become memory T cells, which means that this is great, meaning that the girl that got the first CAR T, what is it now, 14 years later, or 10 years later, she probably has memory T cells that if that tumor ever relapses, those memory T cells come to the rescue and kill the tumor. So we know a few things, but we don't have an iota of PK/PD the way that it exists for small molecules or biologics. And I will try to try to allude to how do we do that. So in the preclinical testing, one thing that is everybody thinks, okay, I'm going to do the preclinical testing, everything is going to look good, and you're going to send Pharmaseed, I don't know how many millions of dollars, what have you. They will show everything is fine, those NSG mice, they will all be great, no death, no adverse effects, everything is going to be good. I'm going to put it into the human, my preclinical is done. Sorry, that's wrong. If you look into the ICH guidelines, and they are totally correct, is that throughout the clinical development, you need to understand your product. If you think that you're going to start a clinical program and it will just run smooth, there will not be any side effects, there will not be any toxicities, let me tell you, you're hallucinating. Starting with the fact that you're giving the drug to a sick patient. Being that you're giving the drug to sick patients, some of your patients will have side effects, will show bad things. Some of your patients may even die. And you're going to ask, is that because of my drug? Or were they sick to begin with? Or you're going to ask, how do I do that? And the regulator will ask you, what's going on here? Patients are dying. You gave your drug, you gave your stem cells, to stroke patients. Half of them died. You're going to say, well, stroke patients die. They say, okay, prove to me that this is not because of your drug. And you're going to have to do that, and this is why your preclinical program has to go on while your clinical program is running. And it's going to have to be in vitro and in vivo. Now, mind you, why in vitro and in vivo? And the reason, first of all, there's cost. But then there's the other thing. At the end of the day, you're producing human products, and you're asking those human products to interact in a predictable fashion in mice, or in a rat, or in a pig. Now, I would, as a veterinarian, I would argue that the xenogeneic hammer in this setting is a 50-kilo hammer, whereas the effects, the pharmacological effects of that drug is maybe a tweak. So do in vitro assessment with patient samples, and I will attest to that, to understand what is relevant and what is irrelevant in your preclinical programs. So you need to be aware, and you have to be prepared for the following. And when you plan your product, when you present your budget to your chief financial officer, you say, I'm going to need to do this, and I'm going to need to do this, and I'm going to need to do this. And like Chaya was saying, he's going to ask you, do you really need to do this? Isn't it dangerous? Actually, you putting a xenogeneic product into a mouse, some mice will die. You don't want mice dying in your safety study. And then you're going to ask, is this part of a global initiative in inflammation and immunology therapeutic area? Because if it is, then you're going to tell them, well, you know what? For this product, I don't need to do that. But I need to understand the mechanism of action of my drug. And you're going to have to fight for that. Because the first thing that the FDA will ask you when they push you to the wall is, do you understand your product? Do you understand how it works? Now, for the most part, we understand how T cells work. But if anybody tells me that they can understand how stem cells work, is it through exosomes? Is that through endosomes? Is that through interaction? Do they engraft? What do they do? We don't understand that. And the FDA will ask you that. Or the other thing is, does it look-see, the discovery phase? Is this something that you just licensed from your University of Pennsylvania, and you say, guess what? I want to know what it does. And in that case, it's a different program, it's a different strategy, it's different tactics. You're going to have to look into existing molecules and data. We talked about the bispecific antibodies. Bispecific antibodies are not all created the same. You have the Fc fragment. You can have a certain Fc fragment on those bispecifics. They will behave differently. Previous animal data. Here's another question. Are you moving into phase 1? Very rarely would you put your product into healthy patients. Or are you moving into phase 1b or phase 2? Do you understand if it is very important, if the patients are sick and you're putting in a product, your cell product will behave very differently than in the healthy animals. Mind you, in Pharmaseed, I think a while back, Itzhak had the program called what was it? Not Young but Relevant. Think about this for a moment. We're taking those mice, they're all 8 to 12 weeks old. They're all on a, vegetarian diet with plenty of exercise, and we're asking them to behave in a predictable manner in the Parkinson's disease or in stroke. And we wonder that they heal like everything is great. Then you go into the patient and nothing happens. Because guess what? Now you got a smoker, obese, diabetic, in their mid-60s. Do you really think that your mouse was predictive for anything? So you're going to have to really think about your clinical development plan. That is, okay, I'll move fast. But this is basically, that's the gist of it. So this basically non-clinical testing for therapies. And this is essentially speaks to the complexity of our product, both in size, both in what we understand that happens to the drug, as well as specifically in cell therapy. The moment that you put a cell into a human, the cell sees adhesion molecules, the cells can be activated. The route of administration will make a very big difference. If you put it into the veins, into the brachiocephalic vein, the first place that those cells will see is the lung. If you put it into the muscle, very different. You put it into a sick muscle, it's very different. Formulation will make a difference. If you put the MSCs in a specific formulation into the arteries or into the veins, some of the reaction may be due to the formulations. And then the disease state. Now, mind you, everybody says, oh, animal models, animal models. Species make a difference. So humans are very poor models for mice. And a sick human is still a very poor model for a healthy human. And depending on the sickness, you're going to have different reactivity. Immunogenicity with the human product will rear its ugly head, even if you use NSG mice. You need to think about cross-species reactivity. That is, once you get the slides and you look for your cells, you look for where your T cells go. You stain for CD3. Is that CD3 cross-reactive with the mouse or not? Very important, availability of orthologous models. That is, can you create the disease? Can you create a like product in the mouse that will give you the answers you need? And sometimes the answer is yes, sometimes the answer is no. As Chaya was saying, your product is your process. If you have to change the process drastically to create that orthologous cell, maybe it's not orthologous anymore. Small differences in receptor binding and the molecular structure of the ligands will result in very different toxicity and efficacy. At the end, you have to think, what is your plan going to do? Again, I will take the privilege of, take the permission of saying something that in Hebrew, Sof ma'aseh b'machshava tchila. You need to know where you want to end. So what do? A way that has worked very well for us, actually at Celgene, is do a lot of in vitro assessment. Collect as many patient samples that you can. Sera, whole blood. See what it does. Take ex vivo assessment from the models and find out in the model what it means. And then, once you have the mechanism of action, once you have the safety, gingerly go very slowly into patients. And then some assumptions and misconceptions. First of all, I would argue that with some exceptions, you really don't need to use non-rodents. Rodents are enough. However, if you're doing local administration, you're looking for local reaction, local distribution of cells, maybe you need something bigger than a rodent. Because 1 millimeter in a rodent versus 1 millimeter in a rabbit versus 1 millimeter in a non-human primate spells very different events. Go into the ICH guidelines. Don't be afraid to engage with the FDA as early as possible. Unlike small molecules, with the Office of OTAT, the Office of Cell and Gene Therapies, the approach is extremely collaborative. So, overall, when you're doing toxicity, demonstrate exposure. Because, if the cells die immediately, and I'm the regulator, so the cells died immediately. What you saw at 61 days is irrelevant. What is the relevance of your chosen species? Mice are your friends, beautifully, because the library of genetic manipulation that you can do with them is enormous. Be ready for follow-up construct. If something doesn't work, another cell. The moment that you have manufacturing changes, transduction methods, you're going to have to do studies to see whether your concept still holds. Time is always the issue. The multiplication of time and money is going to be the bane of your existence. Chronic exposure studies, immunogenicity is a big issue, even in NSG mice. And so you're going to rely heavily on pharmacodynamic models, do a lot of ex vivo analysis of the cells and tissue. My feeling is, almost as a lab animal vet, is that by the end of the experiment, nothing should be left out of the rodent except for the squeal. If you cannot do the assessment, freeze the samples. You will go back to them later when you have side effects. You can take this. I'm sure that Pharmaseed will be happy to share this, and feel free to call me. We can advise you. I can advise you about some of the issues, as long as you're not dealing with myasthenia gravis, which is my project. Make sure that you use both healthy controls in your animal models as well as diseased states, because they will give you different answers, and you're going to have to understand what the answers are. The beauty of healthy controls is that if something bad happens, you know that it's because of your drug. However, being that you're not going to give the cells to a healthy human, you need to also put in disease models. When things go wrong, and things will go wrong, if you don't see efficacy, is the efficacy model dependent? Is it exposure? Did you get exposure? If you have immune modulation, look to the colony status. So in summary, at the end of the day, you're going to have to decide what is your dose. You're going to have to tell your clinical director what is the dose to go with. It's a touchy issue. The FDA asks us to base it on animal studies. I would argue that, especially in the CAR T business, sometimes animal studies are, for dose selection, are totally irrelevant. Questions?
I will start from our Zoom participants. To what degree do you focus on organelles, contact sites in your research?
It depends on the disease. Sometimes they will give you a great answer, and it depends on organelles as well. For tumors, you can get some really nice answers about the tumor microenvironment with organelles. For other systems, there are some companies that will sell you a glomerulus, a kidney, essentially kidney organelle that gives you some good answers. The other issue, the one problem that you have with that is that you pass your cells through the organelle, you don't know that the same kinetics will happen in an animal. So at the end of the day, it depends on the disease, depends on the organelle, depends on the available animal model that you have. The beauty of an organelle is that you remove the xenogeneity issue, you remove the immunogenicity that will happen in a rodent.
Thanks. If you compare a procedure like autologous bone marrow transplantation, that is something that has been approved tens of years, with the you have to administer the patient something to mobilize the stem cells. You take them out, you have to clean them, and then you return to them the cells. That's a procedure approved very long time ago. Now, CAR T cells, it's very similar. I mean, you take T cells. Maybe the difference is that you don't have. You also have to clean it. You have to change the receptor, introduce the receptor, and then turn back it to the patient. So, how these regulatory issues is so much complicated that a procedure very similar that has been approved so long ago.
So the answer is, again, it depends. It depends on your vector, because if your vector is viral, do you know that the only thing you did inside that cells is add that particular receptor? Maybe other things changed as well. Maybe that cell, now that it sees the patient, is going to start, expanding uncontrollably. Take it a step further. If that viral vector has been used already and has been in use, for 10, 5, 3 years, then the burden, the regulatory burden, is going to be much less than if this is a brand new novel transduction methodology that you don't know what happens. So, and on top of it, as already was mentioned, it comes back to risk-benefit approach. If this is an astrocytoma with a life expectancy that is nil, the patient that has everything else was exhausted, then the tolerance of the regulator is going to be very different than if this is, say, even Crohn's disease, which has many other treatment possibilities. So to lump all the transduction methodologies, all the manipulations of a T cell in the same bucket is wrong, because you don't know what else happened during your transduction process.
Thank you very much, Uri. And our next speaker, Nissim Peretz from Itai and Beyond.
Hey. So my presentation is going to be somewhat different than what you've seen so far, because first of all, It's a brand new company, just a few months ago, and I'm gonna present the company and gonna share with you some of our vision. I would like first to thank Shai and Yochai for inviting me to speak. Our company sits in BIOHOUSE, and we definitely enjoy the facilities and the infrastructure that the BIOHOUSE has to offer. So thank you very much. This is a very exciting event for me to for the first time to talk publicly about our startup and to basically uncover our vision. Our story begins, even though our company was established just a few months ago, our story goes way back to almost 5 years ago. When I was a PhD student, I was studying autism, searching for new therapeutic approaches. Some of them were stem cells-based approaches, and that's how I came to the field. And that's when I also met Shmulik. Shmulik was a successful high-tech entrepreneur with 2 children, Leah, a normal teenage girl, and Itai, a 12-year-old boy that was diagnosed with low autism spectrum disorder. Shmulik traveled all around the world to find a treatment for his son. And during his journey, he came across my papers, the papers that I published on stem cells therapy for autism, exosomes therapy for autism, and he contacted me. And 5 years later, we decided to collaborate and to establish a company, an R&D company, that will develop novel diagnostic tools and treatments for autism in a way that we believe that no one has done before. I have to say, a disclaimer, that we are not a stem cell therapy company per se, but we do use the source of stem cells to generate our R&D program and our R&D pipeline and I will explain. So Itai and Beyond is an R&D company that aims to break the barriers of studying and treating autism, and we just, as I said, a new company. And before I will talk about our vision, by the way, I think it's going to be a shorter presentation than what the time that I was given, but I hope to talk as much as I can. So when we talk about autism, so we need to understand what autism is, first of all. And it's somewhat different from other disorders because it's mainly diagnosed by behavioral observation. So the clinician observes the child or the adult, and just by their behavior, he determines and gives the diagnostics of autism. And he observes 3 core symptoms. The first is deficits in social interaction and communication, stereotypical movement and repetitive behaviors, and cognitive rigidity and difficulties in generalizations. Those are the symptoms that if the child is presenting, he is on the labels of the autism spectrum disorder. Autism is now defined as the most widespread psychiatric disorder in the world, with steadily increasing diagnostic rates. If we compare our diagnostic rates just a decade ago, they were doubled. And we're not talking just about overestimation, it is really an increasing phenomenon. And if we think about it, I'm sure that each one of us can think of someone, a relative, a friend that was diagnosed on the autistic spectrum disorder, or have a son or a daughter on the autistic spectrum disorder. And the question is why, for such a widespread phenomenon that can affect every family, that creates a huge burden on the society, there is not a single effective treatment in the world. And believe me, I've done a PhD on it, and Shmulik was traveling all around the world, and there is not a single effective treatment that can ameliorate the core symptoms. There are some treatments like cannabis, for example, that can ameliorate the hyperactivity, for example, but the core symptoms haven't been assessed. When we opened Itai and Beyond, the first thing we did was to try and map and to realize what are the challenges that we are going to face. And I'm still not talking about the challenges of what they talked here, of manufacturing the drug or spreading the treatment. I'm talking about the R&D. How we are going to be different than other companies that put millions of dollars and failed. And we are the small people from Israel. What we are going to do different? And we mapped the challenges, and we came to the foregone conclusion. It's a trivial conclusion that autism is a subcase, a specific subcase, from a much bigger story that we refer as the valley of death. Basically, the long road for the from the preclinical to the clinical trials. And what you talked before, Uri, was very relevant to us because the mice models of autism are not so relevant for the disorder itself. We are trying to mimic autism in mice and rats, and the relevance for the children at the end is almost coincidence. It's not something that you can do. Even if you generate a specific mutation and the mouse displays some kind of a phenotype, this phenotype have nothing to do with what you're going to see in the clinic. So this is a very big problem. And beside of the irrelevance mice model, the insufficient neuromechanistic understanding and the lack of prediction methods of what type of drug will eventually work on the patient itself, on the brain of the patient itself, it's something that is a very big challenge. And eventually we have a variability of patients and variability of phenotypes at the clinics. But when we thought about it, that made us a bit nervous. But the other side of the coin is that the world is changing, and I believe that it's really changing. And new technologies that were not relevant a decade ago are now relevant. Some of those technologies obviously are AI and machine learning for big data, three-dimensional biological tissues that I will talk about, and RNA therapy that came to our life also because of the corona. And what we are doing at Itai and Beyond for the last few months is to create a technology and a pipeline that will combine all of them to generate a model of prediction for drugs that will be, we believe, with higher chances of efficacy in the clinic. We basically decided to establish a platform for scale-up that will allow us to generate a database from clinical tissues, a huge database that will help us create models for predictions of what is going to pass better the valley of death. I can't explain too much about this scale-up platform today because, again, we are a new company and this is ongoing and the IP is still ongoing, but I will explain some of the things that I decided to talk today. We are going to focus in some stage of our development on human brain organoids, and brain organoids were mentioned here. In the field of psychiatric disorders, we believe that brain organoids are definitely the future. For the first time in the history, we can generate a three-dimensional tissue derived from a patient with a structure and proportions of neurons that can mimic somewhat of the patient itself. Not to use the brain of the mouse anymore, but to generate a three-dimensional model of the mouse of the human himself. And if we are talking about the gap between the human brain and the rat brain, we believe that brain organoids stand somewhere in the middle and provide us a new in vitro model that we can study. And people have shown all around the world, brain organoid is a very new, in terms of science, it's a very new technology that is increasingly evolving and always getting better. And they show that they can definitely mimic the most important structures of the human brain. It has greater resemblance to the patient brain. It's enabled cheaper and more successful development, and it's a platform towards personalized medicine to predict what's going to happen to the patient itself, how the patient's brain will react to the treatment. And just recently, this year, for the first time, some lab in Germany created brain organoids with retina. So this is just an example for where the field is going to be. And we believe that if we are going to get to this field now, we are going to be pioneering in this field. And our platform is a scale-up platform that enables us to basically generate multiple numbers of organoids from multiple patients with low cost. Our next step is to collect data from those organoids. Data that will come from high numbers of patients and will basically overshadowing the variability that we're getting in the clinic. Because if we can generate data not from 100 patients, but from 1,000 patients on the autistic spectrum disorder, to create a database with machine learning that we are starting to think about today, we believe that we can predict new targets and new biomarkers that haven't been tried before and haven't yet been failed in the clinic. So this is part of our vision. This is our platform, and our pipeline starts with collecting tissues from patients to generate multiple brain organoids, to generate a database that will help us cluster and do subtypes of the autism spectrum disorders in the population. This will provide us the ability to have new targets for autism that haven't been tried before, to test them again on the specific brain organoid of the patient, and then to understand how the patient is going to react according to its brain organoids, and from then to take the best candidate to the preclinical studies. This is our pipeline, and this is what our company is going to focus within the next few months. So, as I said, we are a new company. This is our winning team. We sit here at the BIOHOUSE, and thank you very much. We're obviously open for collaborations, for questions, for any strategic collaborations and projects. Thank you very much. Yes. Yeah. Yes.
What is the source of your starting material? How do you collect brain tissues from
This
patients?
is the part. Yes, this is the part.
The IP part.
Yeah, this is the IP part that I'm not going to explain. And this is the interesting
but
part.
from living patients, so obviously.
Yeah. This is the interesting part, and that lecture was just a teaser for what we're going to do. But I know that it's a mystery, and I can assure you that it is very interesting mystery. Some of the people that sit here already been exposed to somewhat knowledge, but yeah, we are working on it, and we're gonna have, I believe, we can address this issue once all the.
So you're already working with this. To do that.
Yeah. Mm-hmm. Yes.
Can you discuss some of the relevance of the biology that you're getting from tissue that is derived from iPSCs to make organoids?
So the iPSCs is a part on the process to generate organoids.
Yes.
So
and how relevant, what is the fidelity of that organoid that you get versus taking primary cells from a patient?
So the difference is that when you're talking about the brain, I spent 12 years studying the brain in multiple aspects: electrophysiology, anatomy, morphology, etc. And I can say that there are a few advantages that I found in brain organoids that you cannot achieve in a primary two-dimensional tissue. For example, brain organoids are a developing tissue. You make them develop. And when the structures are being created, you get microcircuits that you cannot get in any other technology, and the proportion of the differentiations of the cells, when you compare it to post-mortem, for example, is the same proportions. And people have shown also, not us, that when you take samples from biopsies of autistic children and you generate organoids, you get what we call alternative excitatory inhibitory imbalance. And excitatory inhibitory imbalance in autism is one of the strongest theories that explain this phenomenon. So it basically, you can get it in the organoids. We are bringing, and it's a long journey, to bring a world expert of brain organoids to the lab. It was a long journey. He did 2 PhDs and 2 postdoc trainings just on brain organoids, and he is working to work in our company. He came from the United States, and because of the corona and because of everything, we are expecting him, and he's supposed to land in our country within a week. So he's going to be in charge on the brain organoid, but just not just the characterization of them, the relevance of them, everything. And we expect that this issue will be asserted. Yes, any question?
I have a question from our Zoom participants. How can you be sure that organoids will cover the whole network of brain centers connectivity that is affected by autistic brain?
Okay, so this is a very good question, and there are 2 answers to this question. First of all, it won't, but it's basically better than other things we have. And the second thing is that today the ability to generate brain organoids from different brain areas, which means that we can generate organoid that will be resembled to the cortex, and another organoid from the same patient that will be resembled to the cerebellum, for example, and to the hippocampus, and then explore each one of them separately. We cannot have a whole brain organoid that cover all the circuitry and the connectivity of the brain, because otherwise it will be just a brain. So we can basically develop just a part of the brain by 5 or 6 different protocols to have different types of brain. And regarding the specificity of autism, this is something that the literature is talking about a lot. In autism is specifically an advantage to use brain organoids because they preserve somewhat of the epigenetic pattern and the protein expression from the patient itself. So we're going to expect to see it on the organoids. Thank you very much.
Thank you very much, Nissim. And our next speaker, we're moving to Natalie Ivgi-Ohana, I'm sorry. Natalie Ivgi-Ohana from Innovia.
Thank you very much. It's a pleasure to be here. Thank you so much, Pharmaseed and Shai, for organizing this event. It's really exciting to be on stage again. Unbelievable how long it has been. I feel like dancing and not presenting, actually. But Shai, you always have a special way to bring me all the way from Haifa to Jerusalem. I used to do that in BIOHOUSE and enjoy some wine. But it is really a pleasure to be here, and congratulations for achieving this. You've heard some of the complexities around cell therapies. The need to control your manufacturing processes to bring very good and strong quality systems into the company. The need to work on right animal models or cell models to prove the proof of concept for the technology, to understand the mechanism of action. You have to have a PK, PD in your animal models. All of that is so complex. I have to tell you, if someone told me all these complexities before I started, I probably wouldn't have been here standing and telling you the story today. But take all of that, multiply that by 1 million, and that's the level of complexity that is really there, or even more, when you are dealing with a new technology that no one have ever done it before. So this is Minovia. We are developing mitochondrial cell therapies. We are the only company actually that are doing transplanting mitochondria in cells. Mitochondria are super important organelles, okay? I think that in the world of cell therapy, these organelles have been overlooked. And what I want to convince you today is that they're very important. I've studied that for over 20 years in my scientific background. First in my PhD in the Hebrew University with Professor Yossi Orli, that taught me that without mitochondria there is no life. And not only because they produce the energy that is required for cells to live, but also because they are responsible for steroid hormones production. There is a great book by Nick Lane called Power, Sex, and Suicide, and it's not a novel. It's a cell about mitochondria. Power is the energy that they produce. This is the ATP. Sex is the steroid hormones, the sexual hormones that are required for creating life, but without them there is really no much sense of living. And suicide is the process of apoptosis that starts in the mitochondria. So life and death in the hands of mitochondria. And that was fascinating for me. When we talk about respiration, we think it's the breathe process, but actually that process takes place inside our cells. This is the mitochondria doing. What are they doing? We feed them with oxygen when we breathe. We feed them with glucose or free fatty acids when we eat. And they take this combination and they produce the ATP, which is the energy going in the cells. They produce CO2 that we inhale, and they produce water. It's the fountain of youth, actually. So all of that is breathing intracellular in the organelle. When we culture cells, and this is important for every cell therapy, when we culture cells, we give them nutrients. We give them oxygen. We culture them in the best conditions. And we think, well, they are alive. They are proliferating. They are differentiating. And who do you think is responsible for all of that? It's the mitochondria. The oxygen given, the glucose in the medium, the free fatty acids will determine if the cells will live or not based on the ATP that will be produced by the mitochondria. Without that there is just no life. But are we checking mitochondrial quality when we are growing cells in culture? Are we testing mitochondrial quality as a quality control for our products, for our cell therapy products? And I'm afraid that very few companies do that. We definitely do. Why? Because we are using mitochondria. This is one. And the second thing is we really believe that that's important. One of the first cell therapies that was ever used is hematopoietic stem cells transplant, of course. And hematopoietic stem cells are very unique. We mobilize them from the bone marrow today using drugs, GCSF or mozobin, and then we can collect them from the peripheral blood. And when we infuse them back, either just as a stem cell transplant or after gene therapy, like some companies do, they actually can home back into the bone marrow, and then they start proliferating and differentiating into multiple cell types, including all the blood cells, including all the immune system, actually. And these cells can circulate throughout the body and reach every organ system. So the thing is with mitochondria, when you think about it, they are super important, but they are different in each stage of this differentiation of the hematopoietic stem cells. For example, when we want them to be stem cells, pluripotent stem cells, mitochondria are silent. Those cells are quiescent, and mitochondria are not active. The cell is producing energy from glucose. It's a very poor energy production process without the mitochondria, of course, but enough if you boost the cells with glucose, they'll get enough of this energy. But when you want them to proliferate, migrate, differentiate into different cell types, then the mitochondria play a very important role. So, if they are healthy, hematopoietic stem cells will differentiate and graft, proliferate, and differentiate. And when they are impaired, we'll get reduction in these activities. So mitochondria for hematopoietic stem cells are very important. What are we doing in Minovia? We are producing mitochondria. Those are the organelles that will give life to the cells. And we are combining them with different cell therapies. And we believe that this is a very, very strong modality. Cell therapies by itself, and with the addition of mitochondria, we actually get enhanced cell therapies. The first product that we are manufacturing is the mitochondria. We produce them from placentas, healthy placentas, and we create a bank of allogeneic mitochondria that can be cryopreserved and later on used. Used for what? We use them to enrich stem cells with mitochondria. There are different cell types that we are focused on, such as hematopoietic stem cells, as I mentioned, autologous, this is our first product. We have allogeneic stem cells in collaboration with Astellas. We are looking into mesenchymal stem cells and CAR T cells. They all require, of course, mitochondria for action. And by this, we are creating multiple cell products that can be used for different indications based on the cell product itself. What we have developed, not only the drug substance, which is the mitochondria themselves, and not only new and novel cell therapy drug products, but we are actually developing our methods. Methods, qualified and very robust methods to be able to measure mitochondrial quality, quantity, activity, not only the drug substance again, but also in the cells after enrichment with mitochondria and in the patients afterwards to measure mitochondrial activity and the efficacy of the therapy. As I mentioned, our lead product are hematopoietic stem cells. We try to keep it simple for the CMC aspect. So if we take the same process as used in the clinic for hematopoietic stem cells transplant, we're actually escaping some of the regulatory aspects that are very, very complex. So we used almost the same process as used in the clinic for many, many years, and we augmented these cells with the allogeneic mitochondria. So we have drug substance that is different and still very, very complex. We have to focus. So if I will tell you about some diseases, for example, Pearson syndrome or Karen-Saron syndrome, Miller-Lee syndrome, you probably never heard of them. Those are ultra-rare diseases genetically caused by mutations or deletions in the mitochondrial DNA. I've never heard of them, even after my 15 years of research in mitochondria. All I knew is that we are all going to suffer mitochondrial disease as we age. We accumulate mutations and deletions in the mitochondrial DNA. We gain dysfunction of the mitochondria, and this is one of the underlying causes to many, many diseases of aging. So when I came with this idea to the first investors and founders of this company, that was the goal in mind. But what we always hear, if we want to succeed, we have to focus. And therefore, we chose the rarest disease of all, I think. This is Pearson syndrome. Pearson syndrome. An ultra-rare disease, only 150 patients in the world. And all I'm going to tell you now is actually not true by the order. Okay, we first went into the patients, which is completely opposite than what you would usually do. And then we went back to try to understand the science. But for you to make some of this make sense, I will go to the science, which was just recently published in Nature Regenerative Medicine, by the way. The first thing we had to show that indeed, you have to know, mitochondria are inherited only from your mothers, okay? We all get mitochondria from our mothers, they pass to our children, and we have tens to hundreds of mitochondria per cell, and in each mitochondrion we have several copies of mitochondrial DNA. It's very unique. You only get DNA in the nucleus and in the mitochondria, and even this is an evolutionary process that continues to happen because this DNA can already be found in the nucleus. Mitochondria used to be bacteria, actually. They entered into the developing cell during the evolutionary process. It is called the endosymbiotic theory, and they transferred most of the DNA to the nucleus. So now the nucleus encodes for more than 1,500 proteins for the mitochondria. They did a great deal, and only 13 proteins are encoded by the mitochondrial DNA. This mitochondrial DNA, if you have a mutation in it, this is lethal, okay, because there is no redundancy. But there are many copies. What does this mean? You can have a state where there is both mutated and normal mitochondrial DNA in the same cell. This is called heteroplasmy. What will determine if there is a disease or not? This is the ratio between the healthy copy numbers and the mutated copy numbers. So all you have to do is to shift the balance towards the healthy copy numbers, and you get a change of phenotype. And this is what Minovia are doing. All the rest is not important. Let's move on. So we had to show that they are actually entering cells. What we did is we isolate mitochondria labeled with GFP, green fluorescent proteins. We incubated them under the right conditions. Well, what are the right conditions? You have to find the right optimal conditions for the cells and the right optimal conditions for the mitochondria. And now you have to find the right optimal conditions for the both of them to live together in the same cell. And that was what Minovia is doing, still under development, by the way. Nothing is optimal. You always want to do better. And you develop methods that will help you to detect this. If you want to create a drug product, you have to show the FDA repeatedly that you are doing the same again and again, robust, qualified, and then validated methods. This is the Oxford nanopore showing the exogenous mitochondrial DNA in the cells. When you increase the dose of mitochondria, you get more and more mitochondrial DNA in the cells. You get more and more content of mitochondrial proteins, and you get better mitochondrial activity. But that's not enough, because what you have to ask is what happens to the cell, because the FDA wants to see a potency assay related to your biological activity that is dose-dependent. Still under development, but what do hematopoietic stem cells know to do? They know to differentiate and to create colonies. We know how to do this in vitro, so we established the colony forming assay as a possible, hopefully, a potency assay for the technology. And what we see is that as we dose these cells with mitochondria, they create more colonies. Potentially, in the future, this could become a great potency assay. Remarkably, these mitochondria not only enter into cells, they are also transferring between cells. So now all your brain starts to move, your brain organoid starts to move, and you say, okay, well, that could be a single treatment. Maybe in the case of a genetic disease, when we have a deletion in the mitochondrial DNA, now we repopulate the cells with healthy and functional mitochondria, with a healthy copy of mitochondrial DNA. Now these cells will go to the bone marrow. They will start proliferating, and they will start differentiating, and will circulate throughout the body and transfer mitochondria. How? Through those nanotubes that you can see here, transferring mitochondria to non-labeled cell that persists over time, more than 4 months in a mouse, okay? In vivo transfer of mitochondria in the mouse. And we can find these cells with transfer of mitochondria in distal organs such as the kidney, not only the blood, and that influences gene expression throughout the kidney. This is actually an aged kidney, which is cool. So we can see a shift from an aged phenotype towards a younger phenotype. That's a completely whole story. Again, focus, Natalie, focus, no aging. We are in rare diseases. And as I told you, we took the other way around. We first treated patients. It was under compassionate use in the rarest disease of all. That is a deadly pediatric disease. There is nothing else. There was only one patient in Israel. And when everyone wants this to happen, it happens. And it had to be the will of everyone around. It was the investors, the company, the parents, the patient, the clinicians, the IRB, the Ministry of Health, to come together around a solution that even if this is not our end product, and it wasn't our end product, it was a syngeneic product derived from the blood of the mother of this patient that very, very rarely is not inheriting the mutation. And we did all of that just to enable the proof of concept for this technology, and it was remarkable. And after that first patient, 3 additional patients were dosed under compassionate use therapy. And later on, we submitted our IND. By that time, I had a company, finally, and some employees in the company and labs. So we can really take this path forward. And under the IND, 5 patients were dosed. And then COVID came, and then everything was even a million times more complex. But overall, until now, we dosed 12 patients. And then we reversed and engineered anything and said, now we have to understand. Now we go back to the models, to the preclinical models. Now I'm waiting for your brain organoids to understand how exactly in a patient can we reverse epilepsy with hematopoietic stem cells. How can we explain that? And that's the kind of phenotype that we observed in our patients. So the path is very, very long. I just skipped 20 other slides, but I think you get the point, and we have really a long path to finalize this process. We did one leap, and I will jump here. I will tell you that, this one. So we said syngeneic mitochondria are non-scalable, and they cannot apply to all the other mitochondrial diseases that are inherited, so we cannot take from the mother. So we switched into the placental-derived mitochondria with this bank of allogeneic mitochondria. Now we can control our drug substance. We manufacture it from single placenta. We can manufacture hundreds of doses of mitochondria for multiple patients, of course, and then we can store them and ship them for the desired point of care, perhaps, or our Minovia facilities. We have both in Kiryat Carmel and in Woburn, outside of Boston. What's remarkable about these placental mitochondria is they are way more potent, okay? They have twice as much copy numbers of mitochondrial DNA per mitochondrion, so we actually need a lower dose to those patients, and they are much more active. Their mitochondrial activity is way more active. That results in more augmented cells with more mitochondria. And this is our scale-up process, which we are now, all of us, digging into, realize all the analytical methods requirements, all the CMC, all the GMP, setting up all the quality systems. So when we go back to the FDA with the initial proof of concept, with some animal data that we did with great CROs also on the way, and with a better, highly reproducible and scalable product, now we're ready to go, not only to Pearson, but also to diseases of aging. Thanks.
Okay, first question from the Zoom participants. Can mitochondrial DNA be transferred internationally? I guess it's
Ah,
internationally from one mitochondria to another.
Yeah, so mitochondria are amazing organelles. They are known for their ability to fuse and fizz. That's what they do in cells. There are two stages of mitochondria. One is fission, which is the breakdown into smaller mitochondria compartments, and the other one is fission, which is the fusion into the network. So mitochondria, not like in the textbook, you don't see them as bacteria spreading, not like even my drawings. It's a network that always mix the content. So mitochondrial DNA as well, once they are internalized into the cells and when they are fusing into the network, they start mixing the content. And when they are transferring mitochondria to neighboring cells, they are of course also transferring this mitochondrial DNA. There is a separate machinery for replication of mitochondrial DNA, of course, that is in the mitochondria, and this is natural, happens in vivo. Yes? Great question. So actually there is no immunogenicity to mitochondria when they are inside cells. The question was, how do we address the immunogenicity to mitochondria? So there is immunogenicity to cells, of course, if they are allogenic. Once mitochondria are intact, there is no immune response against the mitochondria. If mitochondria are ruptured and DNA is released, or formyl peptide that reside in the mitochondria, then you can get a severe immune response, actually. Initially, when we started, we said, let's infuse people with mitochondria. Cells will take the mitochondria up. That was a mistake, because we cannot control what happens to the mitochondria once they are infused. And what we wanted is to really prevent an immune response against these organelles. So once they are inside the cell, there is no risk immunogenicity. For example, when we give blood transfusions, there are different mitochondria from different donors. There is no immune response against the mitochondria. The only risk, though, is the persistence, because we are carrying different mitochondria. Each one of us originally came from different parts of the world, and different parts of the world, there are different haplotypes in the mitochondria, and that means different sequences in the mitochondrial DNA. Minor changes might be very important. We are figuring out what is exactly the match that is required for optimal dosing and for optimal persistence over time.
Okay, one more question from our Zoom participants. Will we all eventually be getting annual placental mitochondria infusions and remain young forever?
We are all going to die. I'm sorry. We are all going to die, but we will die younger. Healthier, sorry. Healthier.
And why not use IPS?
So it's an excellent question. Definitely, I can tell you that when we look in animal models, the results that we get is not only because of the mitochondrial DNA. It's not a viral vector. Okay, it's an active organelle that results in many, many, many cellular activities, and therefore, I agree it could be done by IPS. But if I'll take these IPS cells and I will enrich them with mitochondria, their activity will probably be better and longer lasting. So probably at some day, maybe we will be able to infuse people with mitochondria. Probably they will be needed to be wrapped in liposomes or something and targeted into different organs. But when I finish investor meetings, they all end up and saying, can I get some? So I think we are all going to get some someday, working on it. Thank you.
Thank you very much, Natalie. Our next speaker is my colleague from Pharmaseed, Hadassah.
Okay. Okay. So first, I would like to thank the previous speakers. It was delighting hearing you, and all of you who came here to Jerusalem, and of course online. My name is Adar, and I'm study director in Pharmaseed. And today I will talk with you about cell therapy in the preclinical world. So cell therapy, it's a very sexy term, but why? So the beauty in cell therapy is that it holds the potential to change the course of the disease. So if we look at functional parameter as function of time, for example, it can be the heart beating, or secretion in glands, or metabolism in the liver. We can see that there is 100% of function in healthy tissue. But what happens in case of irreversible damage? So there is deterioration in the function of the tissue until it reaches critical morbidity or even mortality. So cell therapy has the potential at least maintaining the function of the tissue by preventing it from further deterioration, but hopefully it can improve or even restore the function, reaching values that are close to the values of healthy tissue. Unfortunately, we've heard today that there are also many challenges in the area of cell therapy. So even from the stage of in vitro, we need to make some difficult determinations that will affect the whole process later on. For example, we need to choose the cell source. It can be autologous cells, and Chaya, hi, you're already convinced that that is very challenging. We can also use animal cells or human cells for animal models, and we cannot forget the immune effect that it can cause in the animals. We need to choose the cell type. We can use primary cells, stem cells, either adult stem cells or embryonic stem cells, and even more than that. We can use mesenchymal stem cells that were isolated from the bone marrow or from the umbilical cord. We can use differentiated cells or engineered cells. We talked about CAR-T previously. Even after we chose the cell source and the cell type, we still need to determine the cultivation conditions in terms of the appropriate media and growth factors and the harvesting protocol. And in the end of this whole process, we need to make sure that the cells are viable and potent. We need to have an optimal number of cells, and we need to prove that they still maintain their therapeutic potential, and it's not trivial. Now, speaking of potency, here I want to show you an example from an in vitro study we conducted. We show the cytotoxicity effect of effector cells, marked in E, against the target cells, which are cancer cells. The cells were incubated in 3 ratios, 1:1, 1:5 in favor of the effector cells, and 1:20. And here you can see the ratios between the cells. And after this incubation, we monitored after the viability of the cancer cells only. So we see higher cell death of the target cells when we used more effector cells. And that was very important when we moved on from in vitro to in vivo studies. Now, let's talk about this transferring or translation from in vitro to in vivo. In my opinion, for about 15 years of experience, this is where the fun begins. Okay? At least my personal point of view. Why? Because we have a whole set of new challenges. First, we need to consider the biocomplexity of the in vivo model. Think about it. We use live animals and live cells. So the biological noise is very high. We need to consider the specific and non-specific interactions of our cells. The specific interactions are much desired. We want the cells will reach to their targets, leading to efficacy. However, we need to consider also the non-specific interactions, especially with various components in the animal body. For example, the immune system or the circulatory system or various organs. For example, liver, and spleen, and lungs. It means that we need to be able to monitor our cells through the process and also in the endpoints. Okay? We call it biodistribution assays. It also means that we need to be very careful when choosing the cell dosage. On one hand, we need to administer enough cells, allow them to reach the target and yielding to efficacy. But on the other hand, we need to be very careful from their accumulation in various organs, causing toxicity. And here I want to show you an example of biodistribution assay. In this case, we injected to mice 2 cell types, and we quantified their numbers using facts in various organs. For example, spleen, blood, and liver. And in this case, we see accumulation of the cells in the liver. Now, let me share with you a story, personal story. So many years ago, almost once upon a time, we had a big safety project, and part of it was to evaluate the biodistributions of cells in more than a dozen organs. So we encountered few challenges. Okay, it was really challenging. So the first challenge was to choose the appropriate method. We realized very fast that the traditional method using PCR is not appropriate or not suitable in this specific example, in this specific project, because of the specific cell we used in that project. So we had to be expert or become experts in facts. And the second major challenge was regarding the protocol or the cell isolation protocol that needs to be used. Imagine that we need to isolate cells from the blood flow, from the blood, and also from the brain. Do you think it's logical to use the same protocol? No, the answer is no. Okay, but back then we didn't have available protocols. So Meital, who is probably smiling now in Ness Ziona, and also Ilan, were spending days tailoring the appropriate isolating or cell isolation protocol for each and every organ. And this is a story I want to share it with you. So moving back to the challenges in vitro, we need to choose the appropriate animal model. We talked about using in rodents versus using of larger animals and strains. Should it be immunodeficient strain or not? We need to determine what is the appropriate age of the animal and also gender. Male and female, what can we do, are not the same. We need also to determine the administration time of the cell, whether in the acute phase of the disease or in the chronic phase. And we will see an example for that. And of course, to choose the suitable controls, which is not trivial many times in cell therapy. Now, by now, I hope I convinced you already that cell therapy is very challenging. However, we can deal with that challenges having sets of skill and setups to use. For example, we need to have technology and special equipment and facility. We need to have the knowledge and the experience to deal with that or with these challenges. And we also need to work under the philosophy of GLP, because we need to be able to prove that the project was done under the highest standards. So animal models are already used for cell therapy, for example, in the field of cancer diseases, inflammation, vascular diseases, and so on. And now I want to show you some examples from our experience just to demonstrate the challenges that we encounter every day in that aspect. So the first example is in the field of the cancer. Here we monitor the progression of the cancer disease using IVs. And we see that the cell-treated mice were basically cured. That was very happy results, but what were the challenges we faced in this project or in this model? First, we used human cells with mice. We had to consider the immune response of the animals. Another consideration we had to make is what was the disease burden in the time of the administration. Whether we should administer the cells in day 4 in low disease burden, or maybe in high disease burden on day 14. If we were administrating the cells in day 4, we give higher chances for efficacy. But maybe we want to test the cells under the worst case scenario. Okay? We need to choose. The next example is from the ischemia world. We were evaluating the blood flow of mice using laser Doppler. We could see normal circulation of naive mouse and the circulation of an operated mouse that had ischemia in the right leg. We can see that the cell-treated mice had improved blood flow in the right leg over time. We could also see that there wasn't any difference or any significant difference between the cell doses. So for the next project, we used the lower cell dose in order to avoid toxicity. This improvement in the blood flow was accompanied by a creation of new functional blood vessels based on immunostaining. So the last example I want to show you is from the Parkinson's disease model. Here we were monitoring the cognitive properties of the mouse, and we can see similar values of cell-treated mouse with the naive mouse, they are significantly different than the vehicle-treated mouse. This was amazing, but I want to share with you the challenge, or the major challenge in that model. So what is the biggest challenge in Parkinson's disease, and we can generalize that to central nerve diseases? We don't have time, so I can let you know that we need to be able to pass through the BBB, and not the burger chain, is the blood-brain barrier, if it's necessary. So the challenge was, how should we administer the cell? Should it be directly into the blood, or maybe it can be sprayed through the nose, or should it be directly injected to the brain? So the answer is, of course, each and every model should be specifically tailored according to the requirements. So With that, let me conclude this talk. We saw that cell therapy translation is very complex and requires the combinations of skill and setup in order to evaluate the amazing live technology to be applied in the preclinical studies and hopefully later on in the clinical studies and in the clinic itself. So with that, I want to thank you all for your attention. I want to say hi because I promised them to my colleagues in Pharmaseed, and I would appreciate your questions. Thank you very much.
Okay, a question from our Zoom participants. Can you elaborate about points in consideration when it comes to potency assay development?
Yes, good questions. So, when we come to structure potency assay, we need to consider 3 points. First, it's not easy, but we need to know what we want. Okay, this is the major challenge. Okay, this is first. Second, in the level of the product, it needs to be well defined. Okay, it needs to be stable and properly evaluated and so on. And third, it's on the level of the model. The model should be standardized and reproducible. And if we have these 3 points evaluated, we hopefully, or in other words, inshallah, we will succeed. Or we will have a successful potency project. More questions? Galit?
Yes,
Yeah.
we have another one.
Sure.
Can you expand about the CAR T cell therapy targeting solid tumors? Is something different about the standard preclinical models to test this?
It's different. Sorry?
Can you expand about the CAR T cell therapy targeting solid tumors? Is there anything different regarding the standard preclinical models to test this?
Okay. Basically, we have here someone who is specialized in CAR T, so I give him the honor to answer this question.
Thank you, Ada. So, actually, we have to consider basically the basic tumor model, which is actually you have two major type of models. One is the syngeneic models, and the other is the xenogeneic or humanized tumor models. In these tumor models, it is more convenient because you can also to try any tumors, any human tumor, and you can try actually the human product, the CAR T, the human CAR T product. If you would like to use another, for example, syngeneic model, so you will have to produce the mouse parallel, the mouse CAR T cells to be able to target with your technology. But usually people want to check their own technology, usually human CAR T cells. So generally in this type of studies, we used to use immunodeficient mice. It could be SCID mice or nude mice, but when you are going to try CAR T cells, in order to improve the engraftment of the CAR T cells, so we used to go to the more immunodeficient NSG mouse. So in this NSG mouse, you can implant the human tumor and you can test your CAR T cells. Generally, they will engraft very well, and if not, we have other ways to improve the engraftment of the CAR T cells by using some immunosuppressors in addition to the mysis and NSG. Sometimes you need further immunosuppression to maximize the engraftment of the CAR T cells. That's it.
And thank you, Adar. I would like to ask my all the speakers today to join me for the panel of questions. Thank you very much, Natalie, for joining us today. Thank you very much. So, thank you for staying with us until now, and thank you all for your talks. It was very interesting and informative. Now, I do have some questions. I think there are more. Let's start with a more general question. If you were now a researcher at the beginning of your way in research, and I know you are, Nissim, so what was the one tip that you could give to help this researcher, this young researcher, through the process of getting from the idea to the preclinical stage and then forward with the regulations, with your experience to the clinical phases?
Consult the elderly. I think there's a lot of knowledge going around. We just need to know to ask the right questions. We don't have to ask the right people. I think just ask, consult. Our young researcher is very enthusiastic, and usually they are willing to listen to the others. So I think as a good start is just all the questions you have, just ask, consult, and at the end you will find the answer that you're looking for.
You have a lot of experience also.
I would, again, let me go back to Hebrew. Bilan gavoa ani nitla. I hang on those who are way up. I had a mentor at NIH that mentored me, myself, and Itzhak, the founder of Pharmaseed, Irv Kopin. And Irv, he's one of the founders of what we know about Parkinson's, the MPTP model, the whole mitochondrial issue. That's all Irv. And when I first came into his office, he basically said, ask a relevant question. Just ask a relevant question, and once you ask a relevant question and think how to solve it, then everything will fall into place.
So you're both actually saying the same thing. So it's
Yeah.
not about the person that you're asking, it's the correct question.
Yes.
Well, we have a consensus. So, do you think that in the cell therapy niche, cell therapy technologies will stand alone eventually, or will it be required like formulations or scaffolds to get into the clinics? I mean, more than what is used today in the clinic, which is hematopoietic cells or mononuclear cells or stem cells, some stem cells that are approved FDA approved.
Actually, the one beautiful thing about cell therapy, and it depends on the cell obviously, is that depending on the cell, you do get a pleiotropic effect. So chronic diseases, the way I've seen them, chronic diseases rarely, especially for cure, they respond to monotherapy. Even rheumatoid arthritis, that is everybody thinks, well, anti-TNFs respond really well. Very few patients in rheumatoid arthritis respond to one therapy. By the time that the disease is chronic, by the time that the system, that the normal physiology has gone wrong, more than one switch, outside of genetic diseases, more than one switch has been impacted. Because human biology or mammalian biology has got so many redundancies such that once you have a disease, you know that like at least 3 or 4 redundancies have gone wrong. To correct it, to think that you can correct for it just with one switch is overly optimistic. And the beauty of cell therapy is just, you know, even to look at MSCs, look at natural killer cells, look at Tregs. They can do so many things as needed. We hope that they do the right thing. And this is where it boils back to mechanism of action. You have to understand the mechanism of action. So will they stand alone? They have a decent chance, yes.
Nisi, your thoughts maybe?
So the question is if we can use the stem cells therapy alone. And for this, I would take my research experience and the other companies that I was working before. And we see that, for example, and that was one of the questions that I asked about exosomes, for example, that we see, for example, that in a lot of papers, not just one, but thousands, that even if you take the paracrine effect of the cells and you use just it, you don't need in some cases the cells themselves. But my answer theoretically is I believe that stem cells alone and their effects can work by themselves. But after seeing also the complexities really and the regulations, and when you come to the clinic, eventually a lot of the promising things are not being translated. I would be careful. I would be more careful about this statement, but you know.
Let me raise the flag of tissue engineering for a second. Even though I agree with Uri that it has decent chance to succeed, I'm also optimistic of the combination of scaffolds and cells, scaffolds obviously to support the cells, and I've seen some preclinical studies, successful ones, so I'm also optimistic regarding these kinds of therapies.
So the combination, it
Yeah.
could be as monotherapy or in a combination with, for example, scaffolds, formulations. Chaya.
Yeah, people are complex. And the same thing are all the therapies that we're discussing today. So I think there's a lot of things that we don't know yet. And I think that time will give us the answer. But it seems to me that nothing is a standalone. We know now that there's chemicals that you need to give in a specific protocol in order to elaborate a effect or something. So I think the same about cell and gene therapy. I think once we believed, at least when I was young, we thought this is... Thank you for that. So we were sure that this is going to cure the world. I remember us discussing how we're going to change the world. So first of all, I think we learned to be a little bit more modest after 20 years, that we don't have too many products on the field, registered yet. So I think there's a lot yet to learn. We have a long journey. And at the end, it's not one or the other. I think it's going to be a combination of products, technology, different cell, gene therapies together.
I know from my experience that the bystander effect that you have from stem cells, from, let's say, cell therapy or stem cells that you're injecting is very, very crucial, because you can inject the cell and it will get to its target tissue, but then it will be eliminated somehow. But his effect will still be there because of the paracrine secretion, because of activating pathways. So it can be something that can start, like they can live in the body for a very short period of time, but the impact will stay with us for a long time.
I agree, and I think one thing that we started already years ago, but now it's becoming more considerable is the machine learning and the AI. I think there's no way to it. And this is when I speak about collaboration, this is where I mean collaboration. We need to collect the data and we need to share the data, because we're going to learn about these effects only from the totality of the data, only when we will combine the data from different companies, from different products. We do not know today really to say, we know maybe the mode of action. I want to say that we think we know the mode of action, the
Yes.
action. Exosomes, do we really know what they do? We know that they could elevate things.
No, we
they
know
could-
less than what we know. Definitely, definitely.
So I think the more we go forward, we know that we know less. And from my point of view, the real world data, and working together and collecting the data, and using tools of big science will give us, not now, but I think in the future we will get a lot of the answers from ML and AI.
Okay, thank you. Now, moving on to the next question, which is not less important. I know that FDA regulation regarding cell therapy is, as they stated, minimally manipulated, which means that you can take, for example, the whole blood, or you can take the mononuclear layer, the white blood cells. But to go further and characterize specific populations, this still hasn't moved from clinical stage to the market, to a product that you can actually use on patients. So what do you think, since cell therapy is not just mononuclear layer, what do you think will be the next stage that the FDA will approve?
I'll start, but anybody could jump in. The FDA, I think, maybe Uri said it, has one very clear expectation: know your product. And the better you know your product, their expectations are that you need to define your product. Two different companies could come with the same product and with different levels of understanding of the product, and the FDA will then, once they have the data, they will go to the other company and expect them to show the level of later data as the other company gave. And when it's reasonable, not when they just decided to do studies because they had money or time, because most of us do not have money and time. So I really think that FDA makes a lot of sense, and I think it's based on what the industry is going to propose to them. And I think that's also, again, we need to show them a little bit more data, and that's why I think it's not moving forward. I think we're standing in our position and we don't want to move any forward. One thing is not to be afraid. If we think something has a minimal manipulation, we need to state it and then see how it's defined. Maybe the FDA gave us a waiver that they don't need to give us. Maybe we do need to characterize it a little bit more. And these are things that, again, at the end, the FDA is going to get onto it. And they will understand that we have the ability to characterize the cell much better than what we're doing now for minimal manipulations, because manipulated cells. For other products, it's full board.
There's another issue. There's the issue of minimally manipulated, and whether the minimally manipulated tissue or cell in the patient that receives it does the same function originally. And that window, the 360 pathway, that window is slowly closing, as is the 510K. Because what they have realized, the FDA has realized, and this is in part the industry's fault, is that companies, there's a lot of cottage industry out there that uses those particular pathways to do things that they should not be doing. And as a whole industry, at least, I think we need to really weed out those bad players. There is, if you go anywhere in the world right now, there is this cottage industry of stem cells, that people are getting injected for anything that ails you. There is a big cottage industry in the United States, at least for platelet-rich plasma.
You have that in
That
cosmetic
for everything
treatments.
everything that ails you beyond the what is approved. And if we allow that to happen, at some point the FDA will say, okay, guess what, guys, we're going to clean house, and everybody will suffer.
Nissim, Ada, any comments?
No, I agree that eventually I think it's about characterization of your product, and the regulation, as much as it's harsh, it basically just demanding you to know as much as possible what you are doing. This is, I think this is the basic logic of it. So I have nothing more.
So I would like to start with you, Ada, and then each one will answer from his own experience. What do you think are the bottleneck processes? Give me one process moving from your experience, for example, moving from in vitro studies to in vivo studies regarding cell therapy.
Only one.
Choose one. We're short in time.
Choose one. So I think it's, as I mentioned in my talk, I think we need to, in each project, we need to characterize accurately the objective, because we can't have it all. And for later on submissions for the regulatories, we need to have very, very specific. So this is something I advise everyone to do.
So the objective.
Objectives. Accurate objectives.
Accurate objective. Chaya?
What do I think the bottleneck is now? I think I gave a whole presentation about it, but
Just choose one that you
I
think.
really
is
think
the-
that the manufacturing ability of our industry is a bottleneck. If someone that needs now a CAR T treatment, he needs it now. He doesn't need it in 6 months. And usually the patients that we treat now with cell and gene therapy are patients in need. And we need to be able to manufacture the safe and efficacious product. And we need to have a better manufacturing ability.
Uri?
What currently, clearly, manufacturing is a bottleneck. You need to, ask whether wherever you're working with, it ends up almost in a ridiculous situation that you need to reserve the rooms a year in advance, not knowing whether you will have an open IND. But you still need to reserve them, because once you have the open, you assume that you'll have an open IND, you reserve it, and then they put you on hold, and as you call them and say, well, guess what, I'm on hold, it will be another 3 months. And they said no to somebody else. It creates an unwieldy situation. Otherwise, it depends on the indication, depends on, in the development process. I can tell you, if you're going after a really highly unmet need, the FDA is going to be very, very much on your side. They're going to look at that, they're going to look at your data, and they will even tell you. They looked at the data from Pharmaseed, the various pilot studies that we did with a company that will remain nameless, and they said, you don't need to do any additional studies. Go for it. Submit to us the raw data, and we'll let you know.
Thank you. Nissim?
First of all, everything they said, but I will go to the scientific aspect. I think that there is a lot of knowledge, and I would even go backwards more to the basic science. I see a lot of papers that explore exactly the same phenomenon and get to totally different, totally and opposite.
It's the magic judge.
Yeah, and opposite conclusions, and different and the reproducibility, etc. So I think that also before we are getting to the preclinics, etc., we need to gather some kind of a database or something that will do order on everything that is done today, because in the field of stem cells and other fields as well, but in stem cells you see lots of theories, lots of contradicting results, and there's a lot of mess, and you can always justify what you do by finding the right person that supports it, and then when someone opposes you, you can always cite the other ones that, I mean, there is no clear path from what I see of what is going on, and then you go to the clinic like with your theory or your state of mind, and in the back of the mind, you know, it's just one out of others, but I think you can never be sure. And I think that the bottleneck is also in the basic science, like to really say this is not working. For example, I almost don't hear papers that say this theory is not working. If you take this type of cells to this I don't know, some kind of indication, it's not working. You will always find someone to support.
That's why you do sub-analysis.
You do
And then
sub-analysis.
you find a subgroup.
Yeah, I know, but
I agree completely with you. It's a joke.
in the general, I think that the science is it needs to be more restricted also with the methodology, and to organize some kind of established meta-analysis, like or database that can be taken seriously, and to say, okay, I have my conclusions. Let's see if this validated database or something like this support it. I think, for me, this is the...
I think that, in Pharmaseed, we have some really early staged companies that come to us with their results, and they're saying, okay, we have these results in our hands, but we want to see how it works in different hands. And sometimes it works, sometimes it doesn't. But I think this is a very good point to validate your point, to validate your results, and validate the process, the thinking process, the methodology process, and everything that goes behind let's say, the good results that you're showing.
Yeah, totally.
I think
If I can add to that and the preclinical side, because you see investigators and some companies that come to me and for consultation, they say, it worked really, really well when I took it to Indiana in an NSG mouse that has been further transduced to express the human GM-CSF. So, okay, really, the likelihood of that translating then to 10 people with different backgrounds in different clinical centers after, with that degree of noise, if it only works in Indiana, in NSG mice with a specific mutation, yes, then it just but that doesn't bode well for venture capital. So you have to, we need, like you said, we need the journal of negative results.
But it's very complicated. Coming from the preclinical world, it's very complicated because, as I mentioned, every model is really tailored for the specific requirements. For example, we have specific administration time, day 4 and not day 12, or a specific dosage, 6 million of cells and not 12. And each kind of model, it's very, very different. So comparison is really difficult. Okay, so your point is a bit problematic. It's true, I agree with it, but it's a very complicated one.
So thank you all, really. Thank you for joining us today.
Thank you.
Now to the desserts.
I consider it a success. I learned a lot today, and I come with a little bit of knowledge about cell therapy, and still, this is a whole, let's say, huge field of knowledge that even if you're in a small niche inside of it, you have a lot to learn. So thank you very much for enriching us today. And thank you all for coming here today and for joining us online. Thank you.